replication, and DNA modification, among others. Because mm, during the transcription process, all the transcription factors all are proteinaceous in nature. The transcription factor bind to the DNA, then the mRNA will be synthesized. Or you have to know the how they are interacting, how the DNA and protein or transcription factors are interact with this DNA. To study this DNA protein interaction, there is five different types of uh, techniques. One is footprinting assay. This one is southwestern blotting. Southern blotting is for DNA, western for protein, and here we combine this. That is southwestern blotting. That is DNA for DNA protein interaction. And third one is electrophoretic mobility shift assay, and fourth one is hybrid east one hybrid system. Then fifth one is phage display. If we go to the DNA footprinting assay, here DNA footprinting assay, this is a molecular technique used for identification of the protein binding to the region of the DNA molecules. This technique is mainly used to identify the transcription factors that bind to different region of genes such as promoter, enhancer, or silencer. Here in footprinting assay, here the sequencing ladder, and these are two footprints, and this is the control reaction. How this gap we will find when the DNA is treated with the DNase1, which cleaves these DNA molecules, and the if the protein is binding to the DNA molecules like this. You'll see this example here. One DNA molecule is there, and a protein molecule is bind to the DNA. When I add the DNase1, that is a enzyme which cleaves these DNA molecules, but where the protein molecule is bind to the DNA will be not cleaved by this enzyme DNase1. So this gap, you will find this gap. uh, this gap provides you a data that the DNA is here. The band you will not find because due to the not cleavage of this DNase1, and this gap you will find in the gel picture. This is the footprint where we will not find any DNA because protein is bind to the DNA, and DNA will not cleave the DNA. So this is called DNA footprinting assay. But in electrophoretic mobility shift assay, in electrophoretic mobility shift assay is used to detect protein complexes with nucleic acid. In electrophoretic mobility shift assay, if you add the DNA in gel and electrophoresis antibody, and this primary antibody confirms the binding with the protein molecules, and these protein molecules will bind to the DNA molecules, and it will shift with molecular weight, because this molecular weight will change because during the binding process, suppose DNA is 1 kilobase pair and protein is 2 kilobase pair. This is 3 kilobase pair. But after the addition of one primary antibody, it will give rise to 3 kilobase because adding one more primary antibody, and it will shift the molecular weight, and it will give rise to the according molecular weight
Sir,
of the
voice break
DNA
hଉଛି.
primary antibody. Voice break ହଉଛି.
ଆଜ୍ଞା ସାର୍।
Okay. Clear ଆସୁଛି କି voice?
ଏବେ ସାର୍ ଆସିଲା, ମଝିରେ ମଝିରେ ଟିକେ ବ୍ରେକ୍ ହଉଛି। ଏବେ ସାର୍ କ୍ଲିୟର ଶୁଭୁଛି, ପଢାନ୍ତୁ ସାର୍।
ଓକେ, କାରଣ ଏପଟେ ଟିକେ ପ୍ରୋବ୍ଲେମ ଅଛି, କ୍ଲାଇମେଟ ର ବର୍ଷା ବି ହଉଛି, ସେଥିପାଇଁ ବି ମେ ବି ଭଏସ ବ୍ରେକ୍ ହଉଛି। ଏବେ ଆସୁଛି ତ? ଆଚ୍ଛା ଠିକ ଅଛି, ଯୋଉଟା ଆସିବ ଟିକେ କହିବ। In the DNA, the protein will be bound, and this protein will be bound after this electrophoretic mobility or shift electrophoresis. This DNA and protein band you will find after you add one antibody. The shifting of this molecular weight, because protein is bind to the DNA, and one primary antibody also bind to the DNA, and this molecular weight will be shift to a next level. So and this is the difference, that the DNA protein having one molecular weight, and DNA protein and primary antibody will give a new molecular weight. So this shifting, you will find that this DNA is bind to a protein molecules. But in southwestern blotting, here you'll put the DNA inside the gel, and you will pour the oligoprobes uh, to this gel, and these oligoprobes are bind to the appropriate protein molecules, and this form a protein DNA complex, and this protein DNA complex you will easily detect by the UV illuminator or any interconnector dye. And but in yeast one hybrid system, this one hybrid screening in yeast is a powerful method to rapidly identify heterologous transcription factors that can interact with specific regulatory DNA sequence of interest, that is a bait, and this is bait and prey model. This bait and prey model is detected via in vivo reconstitution of transcriptional activators, and this because this is the reporter genes after the binding of the bait. This is positive bait and prey model, like uh, will put a bait to target the prey. Here, like that, in this bait and prey model, detection is based on the interaction of the transcription factor. This is the transcription factor. These transcription factors bind to the this transcription factor. We will call them prey, and with the bait is DNA molecules. This is the DNA molecules. These are transcription factors. This is the prey. This is the bait. Then this bait DNA sequence upstream of the reporter genes. This is the reporter gene. This is the upstream part. And to ensure that the DNA binding result in reporter gene activation. This is this bait and prey uh, interaction is activated this promoter or this reporter gene for further transcription of this uh, DNA molecules. But if you saw the bait, if the bait and prey will not interact with each other, then there is no expression of the this reporter genes or the genes. So you will find that the appropriate transcription factor for this DNA or not. This is the DNA protein interaction part, and the last one is phase display. When a peptide or protein domain is expressed in a bacteriophage capsids. That is, here we will use phage means a bacteriophage. Here you will use the bacteriophage, and this bacteriophage capsid, it is referred um, to as a
ସାର୍
phage display, and it
ସାର୍ ଯୋଉ
huh,
କହିଲେ ପୂର୍ବଟା ଡିବିପି ର ଫୁଲ ଫର୍ମ କଣ? ବେଟା ହଉଛି ହେଲା ଡିଏନଏ ମଲିକୁଲ, ସେ ଡିବିପି ଟା ସାର୍ କଣ?
ଏଇ ଯୋଉ ଡିବିପି ଆଉ ଏଡି ଅଛି, ଏମାନେ ହେଲେ ଗୋଟେ ପ୍ରୋଟିନ ମଲିକୁଲ୍ସ। ଡିବିପି
ଡିବିପି ଗୋଟେ
ଡିଏନଏ ବାଇଣ୍ଡିଂ ପ୍ରୋଟିନ ଆକ ଆମେ କହୁଛେ,
ଆଜ୍ଞା ସାର୍।
ଯାକୁ ଡିଏନଏ ବାଇଣ୍ଡିଂ ପ୍ରୋଟିନ, ସେଥିପାଇଁ ଆମେ ତାକୁ କହୁଛେ ଡିବିପି। ତ ଏଇ ଯୋଉ
ଆଜ୍ଞା
ଡିଏନଏ ବାଇଣ୍ଡିଂ ପ୍ରୋଟିନ ଅଛି, ଏଇ ଡିଏନଏ ବାଇଣ୍ଡିଂ ପ୍ରୋଟିନ ଯଦି ବେଟ୍ ସାଙ୍ଗେ interact କରିବ, then the promoter will activated, and this promoter recruits RNA polymerase to activate the gene expression of the reporter gene. If this DNA binding protein will not react with this prey, sorry, this spray will not react with this bait. Bait is here DNA. If DNA is not binding with this the protein molecules, then it will not recruit the RNA polymerase. Then there is no expression of the reporter genes.
Okay,
Okay.
sir. Thank you, sir.
Here we'll in phase display or DNA binding proteins, it is done by genetic fusion of amino acids. The genetic fusion of amino acid sequence produced due to the clone gene to that code proteins encoded by the phage. Because phage, this is biotinylated oligos or oligo oligoDT nucleotides, and this is the streptavidin coated agarose magnetic beads. Then we will incubate with this phage. Phage will react with this biotinylated oligos, because phage will synthesize its nuclear genome. Then it will say, use this biotinylated oligos, and this biotinylated oligos will synthesize inside this phage molecules, and this phage will display the proteins. This display protein will binding to the this magnetic beads, that is tetrabridine coated magnetic beads, and this phase displaying DNA binding protein specific to the oligos binds, because this is biotinylated oligos. And this enables the display of foreign proteins on a phage code, so as to permit its reaction with other proteins that the phage encounters. This is called phage lead. All the models are to detect the DNA protein interaction. How this DNA and protein interact? Basically, maximum study of the DNA protein interaction during the transcription process, because the enhancer, activator, if you put new activator molecule, some gene is silenced. If you put new activator, this activator is a protein. I will find this. If you know the sequence, target sequence of the DNA, then you will put the injector some protein molecules that binds to DNA, and it will activated or overexpress their silence gene, and it will give a protein product. And this protein product will help to different signaling pathways, and it will cure some types of disease. This model predicts the DNA protein interaction. This is the important topic to know, the DNA protein interaction. Then we'll go to the next technique, that is mass spectroscopy. We also previously described about the mass spectroscopy, how we'll detect by mass spectroscopy of peptide or protein molecules. And in mass spectroscopy, first the proteins from 2D gels or biopsy or biofluids, any biopsy or tissue or biofluids, any biofluids like blood, urine, semen, in protein molecules, you have to isolate that protein molecule. Then you will digest with trypsin enzyme. The trypsin cleaves lysine and arginine residues of the peptide, and it will give different fragments of tryptic peptides. That is, it will cleave the peptide to different fragments by lysine. This is K for lysine and R for arginine residues. This lysine and arginine residues, then you will do the liquid chromatography and mass spectroscopy. In the mass spectroscopy, it will separated according to the charge to mass ratio, and in this charge to mass ratio, the smaller ion moves faster and the larger ion moves slower. By this you will find out the molecular weight of these peptides, and these peptides will hit into the detector machine, and this detector machine, different detecting software is there. It will blast to the NCBI databases, and it will align with this entire NCBI databases to find out which protein having this fragment, which protein having this QGVSLR fragment. Minimum matching should be peptide is greater than 60 to 80%. If the peptide is matching 60 to 80% to the blast P softwares of a protein, then we will predict that this peptide having this protein having this QG peptide will find out the peptide or protein by this mass spectroscopy methods. But how will do this 2D gel, two-dimensional gel electrophoresis? And if we go to the two-dimensional polyacrylamide gel electrophoresis, previous class we have discussed about the SDS page. SDS here, because in SDS page, if the molecular weight of the two protein or peptide molecules is same, it will give a one band. So we could not differentiate that how many proteins are present in that band. So in two-dimensional gel electrophoresis, first dimension we have to do the isoelectric focusing gel, and second dimension we have to do the SDS page. Now, this is the two-dimensional gel electrophoresis workflow. First is we have to do the sample preparation, then first dimension is separated by isoelectric focusing. Then what is isoelectric focusing gel? We have to separate the proteins or peptide molecules according to their isoelectric point, because the protein having same molecular weight but they have different isoelectric point. Isoelectric point means they are charged. Isoelectric point means at which the protein or peptide having equal number of ch- positive charge as well as negative charge molecules. This is called isoelectric focusing point. Every protein or every peptide have different IEF, isoelectric focusing point. Then we have to do the second dimension separation, that is SDS page. Then we will detect the peptide with this process, mass spectrometry process, then image acquisition, then protein excision, digestion, and identification of the protein. In the first dimension, this is the pH strip of the uh, uh, pH strip of the isoelectric focusing gel. This gel having different pH gradient from pH 2 to pH 10. Then in first dimension, we have to add the protein or peptide molecules to this pH strip, that's it, isoelectric focusing strips. And in this strip, according to their isoelectric points, the peptide were separated. Because in SDS page, uh, they are not separated according to their isoelectric point, they are separated according to their molecular weight. If the two proteins having same molecular weight in the SDS page, they will give a one band. But in isoelectric point, if the molecular weight of the two proteins are same but they have different isoelectric point, because they are different amino acid sequence, and it will give different charge to that protein, and it will mm, give different pH gradient. According to the pH gradient, the protein will be separated, that is called isoelectric strips. And this separation of protein then will put into the a second dimension, that is SDS page. Then here we will separate the protein molecules according to their molecular weights. Here we will do not found any overlapping molecular weight proteins. Here the all the proteins are separated according to their molecular weights. In first dimension, they are separated to their according to their isoelectric points. And after separation of these 2D gels, then we'll cut the gels and digest with this tryptic digestion of enzymes from 2D gels, and then we'll separate it according to their mass to charge ratio in a mass spectrophotometer. These are called two-dimensional polyacrylamide gel electrophoresis. Then the next technique is microarray techniques. In the microarray techniques, previously also we have described about this microarray techniques. In microarray techniques, we can detect the protein microarray, DNA microarray, or RNA microarray. We can do any microarray. In the microarray, this is the normal cell. How we differentiate the normal cell and cancerous cell? And this normal cell sample and cancerous sample were grown in a cell culture medium. Then we have isolated the mRNA, and this mRNA in reverse transcriptase process, we have converted to the cDNA, that is complementary DNA, and this cDNA is labeled with green fluorescent probes, and here is labeled with red fluorescent probes. And in the microarray, we have put, suppose we have taken the basic examples, that is p53 proteins. If we here the sequence of the p53 protein is there, if we found that if the red or green, how much red and green are bind to this p53 molecules, that is already present in that microarray, and it will give a signal, a green, red, and fluorescent signal, and this fluorescence amount will be detected by our fluorometer or our detected machine, fluorescent detection machine, and it will give a quantitative amount, how much amount of that protein or mRNA is present. In protein microarray, similarly, we have extracted the proteins, and this protein we have called that this protein with antigen, and this antigen will be labeled with some fluorescence dye. These antigens are labeled with fluorescence dye, and in the antibody chip, this antibody is targeted to this antigen, and here the chip contains the known antibody chip, and this antibody will bind to the targeted antigen, and it will give also a fluorescence signal, and this signal will detect by this fluorescence detector. And like that, we also separate also single samples protein extraction, and this, then this protein will be labeled, and this protein will be hybridized with this protein chip, and this binding will enhance the fluorescence signal, and this fluorescence signal also is detected by this uh, uh, one machine is there, that is called chemiluminescence machine or fluorimeter, to detect this um, fluorescence microarray mRNA or protein samples. Then next one is, what is HapMap type project? If you go to the HapMap, that is haplotype project, was a large scale international research initiative to design or to map genetic variation, because we have to study genetic variation, because due to this genetic variation or genetic polymorphism, our gene sequence will be changes accordingly, and these changes evolutionary process will develop to this the evolutionary human beings or homo sapiens species. To study this genetic variation, what is the change in our gene levels, that we have to do specifically haplotypes in the human genome to help scientists understand the relationship between genetic differences in human health, because nowadays lifestyle factors is the playing major role to cause different types of diseases like diabetes or polycystic ovarian syndrome. These are changes accordingly, their variation is changes slowly, and this will lead to raise to different uh, types of diseases like diabetes will cause a comorbid condition, like it will enhance it will damage the heart, kidney, liver, it will damage anything. So this is the slow poison. To study this type of changes or variation, how the gene is varied, then we have to study the haplotypes, and this was launched in 2002 and completed in 2009, building on the success of the human project, human genome project, because during the human genome project, our gene is total genome is sequenced. So use that sequence to study this variation of all the genes. And in this HapMap project, the objective is to develop a haplotype map of the genome by identifying and cataloging common pattern of the DNA sequence variation, that is haplotypes in different population around the world, because we have to study because here our population having the same gene pool, but in the living in Kashmir or cold climate or hotter climate, they have a different slightly change in their gene pool, because of due to the gene variation due to the climatic change. So this study will be give an example or common pattern, how the gene pattern is previously the gene pattern is there, and then this type of changes in the gene pattern by studying this haplotype map. Then to provide a resource for researcher to identify genetic factors contributing GG, because previously the genome is there, but according to our lifestyle factors we have eat so many different types of junk foods, and these junk foods will changes our DNA by changing variation or polymorphing by these different types of junk foods, and it will give rise to a genetic factors contributing to a disease. And different types of drug response and trade, and we also study how the genes are respond to different types of drugs. Then these haplotypes are specific combination of alleles or sequence variation that are inherited together from single parent. This also transfer from also from single parent to the offspring, and these variation include single nucleotide polymorphism, the most common type of genetic variation in the human genome. Because single nucleotide polymorphism is the major cause of the variation in our genome. Slowly, single nucleotide polymorphism will change our genetic frame or genetic sequences. Then, by studying these haplotypes, researchers could more efficiently identify genetic variation associated with different types of disease without needing to examine every SNP in the genome. Then we have to do the population study. The HapMap project involved individually from diverse population, including African, because we have to study the diverse population, because our gene pool is uh, uh, our gene pool and African gene pool may not be same. There may be some environmental conditions or like this lifestyle factor may change or may cause SNP, and due to this SNP, the variation will be takes place. So we have to do the comparative study of this African population, European population, East Asian population, and South Asian population. And this diversity, then we have to compare this diversity was important for capturing genetic variation that are common in a specific population. uh, we have to do see the if African or European are similar, or African East Asian are similar, or South Asian are similar. We have to do the comparative study. How to sorry how to do this by different pages. Page 1 focusing on mapping SNP in say, individual from poor population, this different population of the uh, this African, European, East Asian, and South Asian population. Then expanded the data set, include additional SNP or and more population. Then page 2 included even more population and extended the data set to provide better coverage of the genome. Because if you study more genome or more population study, and it will give a better result by during the study of the SNP. And this application of this HapMap project is genome-wide association study. The HapMap data has been extensively used in genome-wide association study to identify SNP. Target we have to study SNP, because single nucleotide polymorphism is very danger for gene variation. If you change one nucleotide in a gene sequence, suppose AUG is the start codon. If we mutated the U, that is AG, then the next letter is something, then the gene will not express or translated. So this is the biggest problem of SNP. And then we have to use the personalized medicine by understanding genetic variation. Then we can do by across population. The project led to groundwork for personalized medicine, helping predict individual response to drugs and treatment based on genetic profile. If you know the genetic profile, then we will treat the patients. Then evolutionary study, we can do the evolutionary study, how the uh, mRNA is highly conserved during this evolutionary process. We can study that part by this uh, HapMap. projects, and this is called haplotype project study. Then, if you go to analysis the genome by different techniques, uh, this is the cell, and we have to extract the genetic material first, that is DNA or RNA. Then enriched fragment material, we can enrich or fragmented this DNA or RNA by this DNA enzymes or RNA enzymes or restriction endonucleases. Then this after the breakdown of this DNA, we have to do the overlap. By this overlapping, we can find out the genome sequence of the DNA, or we have to study the expression of this RNA by the microarray protocols. Then, how we analysis of the human genome or human genome project. This is very important because genome analysis of humans, because we have to do the analysis of genome analysis of human to study the genetic variation which causes different types of disease, that is cancer or any Alzheimer's or Parkinson disease or any types of diseases. This genome analysis of human refers to the comprehensive study and interpretation. This is the comprehensive study and interpretation of complete set of genetic materials, that is genome within the human cells. The human genome consisting of about 3 billion base pairs of DNA and contains approximately 20,000 to 25,000 protein coding genes. Only there is 20,000 to 25,000 protein coding genes. And this genome analysis help us understand genetic variation or genetic function and their role in health, disease, and evolution. The main motto is to study the SNP, that which causes the genetic variation which leads to different types of diseases like the diseases like cancer. The cancer will causing by slow process. You do not know when you will, the cancer will evolve. By this mutation process, the cancer will be evolved easily. Then we have to study this analysis of the all the genome where the SNP will occur. Then the key components of this human genome is, first is DNA sequencing. First, we have to sequence the DNA by whole genome sequencing. Previously, we have discussed about this technique. Reads the entire human genome to identify all genetic variation, including single nucleotide polymorphism or any inserted or deleted part of the genome or any structural variants, or that is duplication or deletion or repetition of the nucleotide bases. And if we go to the exome sequencing, exome, intron and exome, only we have targeted exome sequencing because exome are the protein coding region of the genome which constitute about 1%. We have previously discussed only 1% of the total genome, but harbor most of the known disease causing mutation. If the exome is mutated, then any disease will be caused. Basically, maximum uh, cancer, because this is the protein coding gene. If this protein coding genome is mutated, then it will not give the functional protein. The functional protein is required for our different signaling process or hormonal regulation. So this will cause a fatal diseases like cancer. Then targeted sequencing, we also uh, target particular gene. We have to study, like we have previously described, p53 is required for our uh, this is a tumor suppressor gene, and it will restrict the tumor progression or inhibit the tumor progression. To study this, we have to do only targeted sequencing of this particular p53 genome, and we will find any changes or mutation will happen to this gene. Then if mutation or changes will happen to their gene, then it will not inhibited to the uh, uh, cancer cells or cancer tissue, then the cancer progression will occur. Then these are commonly used for diagnostic in known disease related genes like cancer. Then we have to study the genome wide association study, and then genome wide association study the entire genome to identify genetic variation associated with a disease or trait. The trait means one character. It compares the genomes of individual with or without a particular condition to find SNP linked to that disease. Because a single nucleotide polymorphism is very dangerous for our genome. So we have to mainly targeted this SNP. And these study have been essential for understanding complex, like we have previously discussed about the lifestyle disease. One is diabetes, which is very dangerous and fatal in nature, which is a slow poison. Then cancer, then diabetes also leads to the heart disease by identifying different risk variant. And then described about this SNP, and this is the main source where the gene sequence or DNA sequence will be changed by due to this SNP. Then copy number variation, we have previously also described. If the copy number one gene is there and produce X protein, but the X genome is copied or duplicated, then another X will be produced, which is doesn't required for our cell. This overexpression also causes different types of disease. To study this, we have to the regions of the genome that are duplicated or deleted, contributing genetic diversity and sometimes links to disease, that is autism or cancer. Maximum nowadays cancer or autism will happen due to this copy number increases. And the structural variation, that is the large scale rearrangement of DNA, including inversion, inverted. One DNA position is inverted and translocated. Different types of translocation or duplication of this genome, which impact genome stability and also the function. Sorry. And fourth one is functional genomics. The functional genomics aims to understand the role of genes and non-coding regions of the. We always not target to the non-coding regions, because sometimes non-coding region also essential for the different transcription factor binding, or different if the changes in the this non-coding regions, then the splicing properly will not occur and truncated protein will you will find different truncated protein. This will be started by the techniques like RNA sequencing to study the expression and how genetic variation affect the different transcriptome of the gene. Then the epigenomics, the study of the heritable changes in gene function that do not involve changes in DNA sequences, such as DNA methylation. If the gene hypermethylation will occur, then gene will be silenced, or any histones. Histone is a protein. If any post-translational modification occurs, then acetylation or methylation, hypermethylation, hyperacetylation will occur, or ubiquitination will occur. This ubiquitination process degrades the proteins. Then it will not give rise to any functional uh, proteins. And this the degradation of this functional protein or not produce of this functional protein leads to different types of diseases. And this how these genes are regulated and how the environmental factor influence the genetic in expression, like environmental factors or our lifestyle factor. For an example, we go for taking different types of junk foods, and this color or junk foods having the similar structure like our estrogen or progesterone or different hormone like testosterone, like structure, and this they are hydrophobic in nature, and they will easily penetrate through the cell membrane, because cell membrane is a lipid bilayer, and they are also hydrophobic, and it will penetrate to go to the DNA and bind to the DNA. And if our hormone is not required, but it will synthesize that hormone, and this will cause different types of like PCOS, like cancer, overexpression of genes, that is leads to cancer, or PCOS, polycystic ovarian syndrome, doesn't require any hormones, but it will synthesize the hormones, and this is the major problem. Then we have to study, analyze this genome. Then we also study the comparative genomics. This approach compares human genome or genomes with species to understand evolutionary relationship and identify conserved genetic element. It helps in studying human evolution, genetic diversity, and origins of the certain traits. Then this comparative genomic study also help in the study different types of genetic diversity in our genome and origin of new traits by analyzing the human genome. Then what is the application of this human genome? Uh, uh, as we described, the using by personalized medicine, if you use different, if we find the different genetic variant, then if we will use different types of drugs to that individual, and this information also used in pharmacogenomics, that is pharmacology and genomics. Pharmacology means drug research, and genomics to develop personalized treatment plans. Then, for an example, variant genes like CYP to C19 or cytochrome P450. This affect how person metabolizes drugs such as blood thinners or antidepressant. If we uh, check this gene is overexpressed or underexpressed to this different types of drug, because this is very important, cytochrome P450, and it detoxifies the different types of metabolic drugs. Then the disease diagnostic, we can also diagnose, by analyzing human genome, we also diagnose different types of diseases by genetic testing, or hereditary diseases such as cystic fibrosis, Huntington disease, or certain cancer, that is breast cancer, that is mutation of the BRCA1 or BRCA2 gene in our routine. And also genome analysis identify mutation that predispose individuals to specific condition. Maximum study nowadays using this genome analysis study for cancer genomics, to analyze tumor genome analysis to identify mutation driving cancer growth, which proteins are mutated and that drive to growth of this cancer, and provides targets for that personalized treatment like targeted therapy or immunotherapy to particular that protein. Then we have also studied the evolutionary analysis of this human evolution, population, or bacterial evolution, or viral evolution. We can study by this genome analysis study. And in population genomics, then we'll large scale genome projects like 1,000 genome projects or human genome diversity project, mapped genetic diversity across different global population, helping to identify genetic variants that contribute to the human diversity and different types of disease risk. And this is the important prenatal or neonatal screening. Nowadays, prenatal or neonatal screening is there for testing any mutation occurs in our genome, for testing our Down syndrome or Patau or Edward syndrome during this prenatal or neonatal screening. And this genome analysis increasingly used to parental testing, that is non-invasive parental testing, that is NIPT, to detect chromosomal abnormalities like Down syndrome, or newborn screening. Early genome analysis also of newborns can identify genetic disorders like metabolic diseases, allowing for early intervention and different types of treatments. And some major human genome projects are: first is human genome project, that
Particularly,
is,
sir.
yeah.
ସାର୍ ଯୋଉ ପ୍ରି ନାଟାଲ ରେ ଯୋଉ ଟିଫା ସ୍କାନିଂ କରନ୍ତି ସେଗୁଡାକରେ କଣ ଏଇ ଏଗୁଡାକ ଆବନର୍ମାଲିଟି ଗୁଡାକ ଦେଖନ୍ତି ନା
ହୁଁ
ଆଉ
ହୁଁ
ଅଲଗା କଣ?
ନାଇଁ 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 ପ୍ରି ନାଟାଲରେ ଏଇ ଯୋଉ ସବୁ ସ୍କାନିଂ କରନ୍ତି, ଜେନେଟିକ
ଆଜ୍ଞା
ଟେଷ୍ଟିଂ ସ୍କାନିଂ କରନ୍ତି, ଏଗୁଡାକ ଟେଷ୍ଟ କରିବା ପାଇଁ କି କରନ୍ତି ସେମାନେ। ବେସିକାଲି ଦେଖନ୍ତି କୋଉ କୋଉ ଜିନରେ ମ୍ୟୁଟେସନ ହେଇଛି କି ସେଇ ଜିନରେ କଣ ଚେଞ୍ଜେସ ହେଇଛି, ଯୋଉଟା କି ମେଜର କି ଡିଫରେଣ୍ଟ ଟାଇପ୍ସ ଅଫ ସିଣ୍ଡ୍ରୋମ କେसेस ଜାଣିବା ପାଇଁ, ଦି କ୍ୟାରେକ୍ଟର ମେଜର ପ୍ରୋବ୍ଲେମ ଦି ସିଣ୍ଡ୍ରୋମ କେसेस।
यस सर।
And, nowadays these are doctors are using every medical using this prenatal or neonatal screening. And this is the major human genome projects, that is a human genome project that is completed in 2003, and this was a landmark because we know all the genome sequence of the DNA, and an international research project that is sequence the entire genome and it will provide fast complete map of the human genome, laying the foundation of all subsequent genome analysis or work. This is the foundation. Then the second project is thousand genome project. This is the also a international effort that aim to sequence the genomes of over 25,000 individuals from different various population for comparison, how the genetic variation of these different population of worldwide, to providing a comprehensive map of human genetic variation. Then this we have discussed about this HapMap project that is focused on cataloging common genetic variation or haplotypes and their association with diseases, aiding researcher in studying how genetic differences affect health. And fourth one is the Human Cell Atlas, that is an ongoing global initiative to map every cell type of in the human body using single cell. Nowadays, researchers using single cell genomics and single cell proteomics or single cell transcriptomics to study the any cellular diversity or how genetic variation influences cell function. Because if we know the cell functions, then we will go for the tissue, then we will go for then organism. This is the major human genome projects study. Then we go for the next one, that is worldwide affected by this Plasmodium falciparum. This genome also analyzed, and this genome analysis of Plasmodium falciparum parasite responsible for the most severe form of malaria in humans is essential for understanding its biology, evolution, how this species increased by this drug resistance, and mechanism of the pathogenesis also increases. And this Plasmodium falciparum genome was sequenced as a part of global effort to combat malaria, providing insight into potential therapeutic targets or different types of vaccine development against this falciparum genome. Then, if you go to the genome size of this Plasmodium falciparum species, this genome is relatively very small and comparatively 23.3 million base pair, and it is organized into 14 chromosome and 5,300 protein coding genes. And this genome, Plasmodium falciparum, is extremely adenine and thymine rich with an overall 80 content of about 80 to 90%. This makes it challenging for traditional sequencing method, but it also contributes to the parasite's unique genetic structure and regulatory mechanism by this adenine and thymine molecules. And these gene families, Plasmodium families, that involve in the immune evasion in the host cell, as we previously described about this Bayer gene. There is different types of Bayer genes, and this gene, if it changes by the variation, and this will encode different types of Plasmodium falciparum erythrocyte membrane protein 1, which mediated the cytoadherence and immune evasion by this enabling the parasite to avoid immune detection. If we changes the sequence of these Bayer genes, if the if our immune system first produce this Bayer one, suppose for an example Bayer one, But if it changes in the second time of infection, the gene will be changes to var4 or variable 4 gene. And if the expression of the variable 4, then our cell will next secrete the antibody against this var4. But during the third time infection, it also changes to var27 or 15. Then these changes uh, helps the parasite to avoid our immune detection. And the different types of also different types of disease genes like refin, staver genes or clac or mfc genes. These are also analyzed, and these genes undergo antigenic variation due to their different types of switching of these bare genes. uh, and this switching of this bare gene causes antigenic variation and allowing the parasite to avoid detection of the host immune system, contributing to its ability to cause chronic infection. And because during the repeatedly bite of this plasmodium species, these variants also changes due to this antigenic variation. Our immune system will cannot do anything to this variable change. And also due to these variable changes, and our drug also resistant to that genes. And the genome analysis also identified gene is associated with drug resistance. The major challenge in malaria treatment, that is this pfcrt mutation of the genes are linked to resistant to the chloroquine. If this gene is mutated, that is resistant to the chloroquine, which is the commonly used drug to treat malaria. Then this PFMDR1 and DHFR and DHPS and K13, these also resistant genes of this Plasmodium species. And if we go to the genome plasticity and evolution, then the Plasmodium falciparum genome exhibits genetic plasticity that allows the parasite to rapidly adapt to changing environmental conditions, including drug pressure or immune system challenges. And this adaptability is driven by gene duplication, mutation, and recombination of these different genes. And this recombination and gene conversion between the members of multi-gene families contribute to antigenic diversity and immune evasion of these parasites. Then what is the benefit of this Plasmodium falciparum analysis? Na- we have to target the different drug discovery and development. We have to develop different drug during this analysis of this Plasmodium falciparum genome. And this drug targets is a key metabolic pathway of this parasite, such as its folate biosynthesis or apicoplast, a plastid organelle for which is essential for the parasite survival but absent in humans. And we have to target this because if we target different metabolic pathways to the parasites, if our similar with our parasite, our metabolic pathway, then our metabolic pathway will be also harmful. So this also help for only find out they have folate biosynthesis pathway and apicoblast pathway. These are only in parasite. Then we have to target this pathway. Then we have to do the vaccine development against these genes, and the PFM1 has been critical because due to their variation or antigenic variation of this gene, we cannot do any vaccine like treatment. Then genome data are used to identify conserved region of the protein involved in invasion or immune invasion that could be targeted by, because some proteins have their conserved region. They have hierarchically transmitted from parents to the offspring. If we target that part, then it will be develop a positive vaccine treatment. Then with the comparative genomics, then we use different Plasmodium species of Plasmodium falciparum with other malaria causing parasite like Plasmodium vivax or Plasmodium knowledge provides insight into evolutionary difference, how they are different, and their evolutionary changes, how their gene pool is changes. It also highlight unique genes and pathways that could be targeted or treatment of prevention. Then population, we have to study the population in during the changes uh, during this Plasmodium falciparum infection, how their population gene pool also changed during this malaria infection. We have to study this population genetic, and the study of genetic variation across the population help to understand epidemiology of malaria and can guide strategy for eradication and control. And if you know the host parasite interaction, we also study the host parasitic interaction analysis by this that genome study. And this host parasite interaction study uh, with human host with inverted blood cell, because they are infected with the red blood cell and causes anemic condition, and the patient will be die due to the lower take of these oxygen molecules. And this identifying gene involved in erythrocyte invasion, that is MSP, as previously we discussed about this merocyte surface protein that help develop therapeutic intervention that blocks the parasite entry into the host cells. And these are the major genome project related to this Plasmodium species, and these are Plasmodium species falciparum genome sequencing project. It also completed in 2002, and this is the first providing detailed map of the parasitic genetic material and malaria gene, an ongoing global network for analyzing genetic data of this Plasmodium falciparum species. And this different PF3K project is going on. And welcome Sanger Institute Malaria Program, because uh, this institute has been instrumental in sequencing in only targeted by Plasmodium species and other malaria parasite genome to support research in drug development and disease control. They have only targeted for drug development against this Plasmodium species. Then, if we go to the next one, that is genome analysis of Mycobacterium tuberculosis. Then, this genome analysis by Mycobacterium tuberculosis responsible for tuberculosis, as we know, TB, that is a very life-threatening disease, and this has been instrumental in understanding its pathogenesis, drug resistance mechanism, because TB patient have some drug resistance mechanism, and the and potential target for treatment of and vaccines. The sequencing of this genome of tuberculosis was landmark achievement, providing insight into how the pathogen caused disease and how it will be controlled. And if you go to the genome size of this TB tuberculosis species, it is approximately 4.41 million base pair, and it contains 4,000 genes, and that encodes proteins and representing relatively large number genome of the bacteria. As previously we have discussed about Plasmodium falciparum, that is 80 content is high. Here the GC content is high, that is 65%, which influences its coding potential and contributes to its unique physiological traits. And they have very virulence genes, and these tuberculosis harbor several virulence factors that allow it to survive and replicate within the host macrophages, because very dangerous disease. They have integrated inside the host macrophages cell, and these host macrophages is our primary defense system. And if it enters with the host macrophages, then our innate immunity will not work. Then this key virulence gene include ES86, and these are ESX1 secretion system. This is the critical for modulating host immune response for enabling the intercellular survival, and the PPP gene family. This also causes antigenic variation and immune evasion mechanism, and this RT1, a key genomic region involving virulence factors like absent in the vaccine strain like M. bovis BCG, but present in different types of tuberculosis genes. And this genome also provides the lipid metabolism genes, and this Mycobacterium tuberculosis is a known for unique complex cell wall and contains mycolic acid, which contribute to its resistance to desiccation and antibiotics. And these genes are involved in lipid metabolism, such as fatty acid synthesis and polypeptide synthesis, that are essential for synthesis of microbacterial cell wall, because the MMPL gene family is also involved in transporting lipid across the cell wall. And also this genome analysis provides the different types of dormancy and latency of this microbacterial tuberculosis gene, that is DOSR regulon or LAC1, LCL1 and LCL2. And some drug resistance genes like antibiotic resistance genes like, that is CATG. This mutation CATG lead to the resistance isoniazid first line of anti-TB drugs, and RPOB. This is also resistant to rifampicin, and PNCA and EMBB. These are the GYRA and GYRB. These are also called the drug resistance gene. If you put any drug and this gene is activated, and it will throw out the drug to outside of the cells, and uh, the microbacterium species will not die. Then this analysis will help to target different drug development, new drug development for this drug resistance bacteria, and uh, on, and know the understanding of the drug. We have to understand the drug resistant, different drug resistant, multi drug resistant genes, and this multi drug resistant gene work as efflux mechanism. Efflux mechanism means it will if the drug will go inside the macrophages cells, and they will activated this multi-drug resistance gene, and this gene will throw out from inner side to the outer side of the drug. Then the drug will not act on this tuberculosis species. And this study also analysis of the different vaccine treatment to the this tuberculosis patient, that is BCG vaccine used for TB prevention, and evaluation of major transmission study. Genome analysis has been a crucial understanding in the evolutionary history of this tuberculosis species, showing it has co-evolved with human over millennia. This co-evolve any disease which is co-evolve with human is very dangerous, and this comparative genomics has revealed that the basic lineage of this tuberculosis is associated with higher drug resistance rates and is more virulent in certain population. And the phylogenetics and whole genome sequencing of this uh, species allow researchers to trace the transmission pathway of the TB within the community and across borders, aiding in public health intervention. By diagnostic, after analysis of the genome, the molecular diagnostics such as genetics expert MDB RIF test, which is rapidly detects the presence of this tuberculosis DNA, and whole genome sequencing also used for profiling of this TB microbacterium tuberculosis species. And here some genome projects initiative of this tuberculosis, that is the tuberculosis genome project, is the one fast effort to sequence this tuberculosis and completed in 1998, providing a comprehensive map of the bacterium genetic material. Then TBARC, that is tuberculosis antimicrobial resistant consortium, and cryptic, that is comprehensive resistant prediction of tuberculosis in international consortium, and WHO. This is the major global TB program. Today also use this program, that is WHO global TB program. WHO initial focus using genomic data to track spread of this tuberculosis species, and particularly drug resistant strain. Nowadays, TB is very dangerous, having these drug resistant strains, and guide public health policies. Different policies are also, after analysis of this genome, different policies also implicated in different regions or different countries. Then after all the genome analysis, then how we manipulate the genomes by site-directed mutagenesis. What is site-directed mutagenesis? Then site-directed mutagenesis SDM is a molecular biology technique that involves making specific changes to a particular genes or DNA sequence. It is used to study the structure and function of proteins by introducing mutation at the targeted site. If some species or the like previously studied, the particular gene is suppose var genes. This var genes is suppose antigenic variation will take place. If we modify this gene or any changes in particular var gene, then it will be helpful for our treatment process. How this SDM work? Now, if we give this is a plasmid cloning vectors. This plasmid is for in bacteria, and then we inserted the gene of targets, and then strand separation by primer with here we put the primer. The strand separation will take place by heating. Then these two strand will be separated. Then we will put the primer against the desired mutated sequence. We have to choose the primer, mutated primer for this gene, particular genes. Then this mutated primer strands synthesized by this DNA polymerase and ligase, and this one is original gene, and this gene is sorry sorry this is the synthesized gene, normal gene, and this is the mutated gene by using this mutated primer sequence. And then we digest wild type DNA, transform bacteria, and generate the daughter cell, that is mutated cell. And this mutated DNA can used for the treatment of the various genomes. And this SDM can be used to add, delete, or replace specific sequence in the particular genes or codon in a DNA molecule. And this allow researcher to manipulate specific amino acid in a protein. If we can use manipulation techniques to use different code, change the codon sequence. And the application of this site-directed mutagenesis used to study the resulting translational product for protein engineering, functional characterization, and modeling of mutation found in clinical sample. And comparison to general mutagenesis, SDM differs from general mutagenesis, which uses mutagenic compounds or high energy radiation to randomly alter the DNA levels. Then this technique and kits, and there are many techniques and commercial kits for generating point mutation in this particular genes or short addition or deletion. This limitation of this SDM is most of these method involve multiple step and are limited by the size of insertion or deletion, because we cannot insert a large fragment. We can use a small fragment or small insertion or deletion a particular gene, not a targeted to a large genome. Then next one is the how we'll know the desired DNA or gene which will be transcribed to mRNA, and mRNA will give rise to protein. After the transcription process, the promoter will after the transcription initiation, it will first synthesize the reporter gene, and this reporter gene give an product, that is enzyme. If we add the substrate to a particular solution, then it will give to a product. That product will quantify by the luminescence or fluorescence, and then we'll know that this promoter, this genome is now synthesized to give rise to one protein. This can reporter gene used during the synthesis of insulin, because previously insulin is used for the royal family in London. They have used it by pancreatic extract. Nowadays, a researcher do using these reporter genes to know how the insulin is synthesized or not in a bacterial cell. Because bacteria do not know, we have inserted the DNA insulin gene to the bacterial genome, they will synthesize the insulin. If bacteria know that this gene is not required for me, then it will put into the inclusion body, or not like a insulin protein, then the insulin protein is not properly synthesized. So researcher put the reporter gene in a particular places before our gene is synthesized. Then the temporal or site specific expression, this is the normal expression of our genome. The genomic DNA is replicated. Then they have promoter, different exon and intron boundary. These are exons are protein coding region, and these are introns are not protein coding regions, and these are transcription by RNA polymerase. And this is the primary transcript which contains exon intron boundary. After the splicing, the introns, that is non-coding protein regions, were removed, and the mature transcript contain different exon 1, exon 2, exon 3, exon 4 complex, and this mature mRNA then translated to a functional protein. These are the normal cases. But during the gene silencing, how the gene will be silenced or transcribed, sorry, how this gene or exon is silenced, using by two method. One is miRNA, that is RNA interference genes, and another one is siRNA, that is small interfering RNA. I will put, as we for an example, nowadays tomato, the hybrid tomato uh, will not degraded by the enzyme. The enzyme doesn't digest the new hybrid tomato, because you, by using this miRNA or interfering microRNA, it will target that particular gene which ripen the tomato. We use that miRNA to that particular enzyme. That enzyme will be digested by this microRNA, and the tomato will be ripened slowly. How this gene silencing will be takes place? By using this microRNA. This microRNA is synthesized by our own DNA, and this regulated the genes by the gene will be overexpressed or underexpressed or a basal level by this microRNA. And this microRNA having the a loop like structure, and this is a hairpin like loop like structure, and synthesized inside the nucleus. And in the nucleus, the DROSA uh, cleaves the microRNA's overhanging regions, and these are the overhanging tails, and it will cut the overhanging tails, and this signal gives to export this microRNA to the cytoplasm. Because microRNA, this microRNA target to the mRNA, because mRNA codes for proteins. If we digest the mRNA, target mRNA, it will not code for any proteins, and the gene will be silenced. And this microRNA export to the cytoplasm, because mRNA are present inside the cytoplasm. This microRNA then unwind by microRNA dicer protein, and this dicer will unwind the microRNA, and this unwind of the microRNA form a asymmetric Rees complex. This is the Rees complex, this RNA inducing silencing complex by this microRNA, and this is complementary to a particular gene. That gene having this particular complementary sequence, and this mRNA having complementary sequence bind to the microRNA, and this microRNA is form a complex, that is this This is RNA inducing silencing complex. During the ribosome process for synthesis of the protein, this RISC complex will inhibit and do not form any protein, and this is called translational repression, means do not produce any protein product. But siRNA is will give outside from the cell. This is the lab synthesized method. This is in vitro synthesis. This is not synthesized inside the cell. We have to inject it from outside to target a particular gene. We have to know the by human genome projects, we know the sequence of the gene or DNA. Then we'll synthesize a double-stranded RNA of double-stranded RNA complex, and this complex is complementary to our target gene or mRNA. And then this will inject to the cell of the injected to the cell. Inside the cell, the dicer protein digests the siRNA duplex to form a complementary RISC complex, and this RISC is RNA inducing silencing complex. This will target to a particular mRNA, and it will complementary to the uh, target mRNA, and this is it will cleave the mRNA, and the protein will be not synthesized, and this is called gene silencing by translational repression. Then after gene silencing, how we can do gene silencing by gene knockout or gene knocking. And in gene knockout and gene knock-ins are two powerful genetic engineering techniques used to study gene functions by altering the genome of an organism. These methods allow scientists to either inactivate it. Inactivated means knockout of a particular gene, removal of that gene, or introduce a particular gene. This is knock-in inside a DNA sequence to understand the role of specific gene as previously described. Gene knockout method. Gene knockout involves in removing or inactivating. Knockout means out. Gene is removed to study the resulting phenotypes, which helps in determine the function of the gene. This method can be used using various techniques like lattice techniques, that is CRISPR-Cas9 models. And these key step of this knockout is target identification, or which gene of interest should be selected or for knockout. Then the design for CRISPR-Cas9 or nucleases, a guide RNA. Like this guide RNA, this microRNA is a guide RNA. It will bind to complementary sequence to a particular genes for repression, or binding to this mRNA for repression. Like this guide RNA specific to the target gene sequence is designed. This guide RNA having the sequence similarity, complementary sequence similar to the target gene, then it will then it causes a double stranded break. This Cas9 is called an enzyme. This induces a double stranded break. It will cut the double stranded of the target gene, and and after cutting, our DNA repair system is there. The repair is non-homologous end joining. This DNA repair mechanism frequently result insertion or deletion, causing a frame shift mutation, and this frame shift mutation effectively knock out the gene, knock out the target gene. Similarly, in gene knock-in method, gene knock-in is a process of inserting a gene into a specific location of the genome. This method is used to introduce a new function or correct a defective genes. In this step, first gene selected, we have to select a particular gene of interest, then homology directed repair. This is the repair mechanism, that is after double stranded break is induced at the target site. Also like this, first we have to break the DNA molecules, and then donor DNA template, like this. If you break these DNA molecules, then we have to insert a new, like this gene of interest, and then this gene of interest uh, carrying the desired gene that is inserted, and this gene integration, this cell repairs the break using donor DNA template, thereby incorporating and the new gene at the specific locations. And this, and what is the difference between this knockout? Knockout means removes or inactivated a gene to see the loss of function, and knock-in is introduced a new or modified genes to study the gain of function or repair or defect of the particular gene. Then both methods are widely used in genetics or drug development or therapeutic research. Then the last one is the what is the benefits of this proteome analysis? Uh, in the proteome analysis, it involves a large-scale study of proteins expressed of an organism, tissue, or cell under specific condition. Because protein always secreted when the cell required and or organisms requirement. And this proteomics is the field of research focused on understanding the structure, function, and interaction of entire set of the proteome, entire protein, all the proteins in a given biological context. And this proteomics plays a key role in understanding cellular processes, disease mechanism, and also biomarker discovery. In this proteome technique, we previously also described about the mass spectroscopy, which is a study in proteomics, or tandem mass spectroscopy. We use a sequencing a protein by fragmenting peptides, like we have discussed about, like you using trypsin enzyme to fragment different peptides, and or isotope labeling, like Silac or TMT, or I track different isotope labeling to particular amino acids to know whether this protein is introduced or where this protein will go. It will present inside the nucleus or cytoplasm or any other organelle or extracellular matrix by this tandem mass spectroscopy or isotope labeling. We also describe about two-dimensional gel electrophoresis. We separated the proteins according to their in our first dimension. We are separated the protein there according to their isoelectric point. In the second dimension, we separated to their charge to mass ratio, and then we do the proteomics analysis. We also describe about the different protein microarrays assay, and liquid chromatography and mass spectroscopy analysis. And this MSMS also modern approach. For first we have to separate the proteins according to liquid chromatography techniques, then we will separate it with this mass spectroscopy approach. And this surface plasmon resonance also SPR detects protein-protein interaction in real time. Previously also we have discussed DNA protein interaction or protein-protein interaction in real time by measuring the change in refractive index when protein bind to a specific ligands immobilized on a sensor or chip. This technique is valuable for studying protein kinetics or binding ability. This is for this protein-protein interaction is important for our say, receptor or ligand signaling for study. This will be studied by this surface plasmon resonance. And the X-ray crystallography and NR spectroscopy used for study the protein structure, how the structure primary, secondary, and tertiary quaternary structure will be found. And we also describe about different bioinformatics tools like protein databases like UniProt, Proteomics DB, and Pride store for protein sequence and functional annotation. And this is the important protein identification software, that is Mascot, Sequest, or MaxQuant, processes for mass spectrometry or data to match peptides to known proteins. We also study the protein-protein interaction network by analyzing using string. Uh, Yesterday we have also described in string how they are interacting, how their pathway are formed to understand the functional relationship among the different proteins. Then, what is the benefits of this all proteomics applications? And if we go to the biomarker discovery, proteomics plays a crucial role in identifying biomarkers for different diseases. By comparing these proteomes of healthy and diseased tissue, researcher can identify protein that serve as a diagnostic or prognostic indicators for conditions different like cancer, cardiovascular disease, and neurodegenerative disorders. And for example, cancer proteomics has led to identify nowadays this cancer proteomics is increasing to lead to identify proteins associated with tumor growth. If we analyze the proteins, all the proteins of the tumor growth, which protein is overexpressed, then we can target that particular protein or metastasis. Metastasis means travel of the tumor cell to different organs. Like from breast cancer, it metastasis to colon or liver or kidney to different organs. And drug resistant, and which protein is overexpressed during the treatment of drug by this cancer proteomics, and this offering a target for therapeutic intervention. Then we also by studying proteomics, we also uh, synthesize different types of drug development, and this proteomics aids in drug target identification by revealing proteomics that are differentially expressed or modified in disease, and it helps identify drug binding site, understand drug effects, and assess of different targets effects. And proteomics is also used in pharmacoproteomics, which is study how individual proteins affect different drug response, which contributing to the personalized medicine. And we also know the understand different type of infectious disease mechanism, and we also study the post-translational modification by epigenomics or epiproteomics by how these protein are post-translationally modification by phosphorylation, glycosylation, ubiquitination, because overexpression of this ubiquitination protein will degrade all the proteins. So how this ubiquitination is overexpressed or underexpressed by this proteomic study. And in phosphoproteomics, because phosphorylation is dephosphorylation for activation of protein or enzymes by this phosphorylation and dephosphorylation kinase enzyme. If the phosphorylation, over phosphorylation, or under phosphorylation can be studied by this proteomic study. And we also study the protein-protein interaction. Previously, we also saw the different examples how this protein-protein will interact with each other. This is called interactomics, and in proteomics, we also use as key components for system biology, and it integrate genomics, transcriptomics, and all the metabolomics to provide a holistic view of different biological processes. And this proteomic data can be used for model for different cellular pathways. We can target different cellular pathway. If we know the which cellular pathway is causing cancer, then we can target that cellular pathway and understand how changes at the protein level uh, that affect the entire biological system. And also this proteomic study also helping in agricultural biotechnology. In plant and agricultural proteomics, this is used to understand how plants respond to different environmental stress, drought. In drought condition, which protein is overexpressed or underexpressed. In heat condition or pathogen interaction with the plant, which protein is over or underexpressed. This knowledge can guide the development of more resilient crop varieties. And also in biotechnology, proteomics is applied in the production of different recombinant proteins or enzymes or industrial byproducts. Or this is the important for nowadays. They are targeting the vaccine for use for vaccine development. This proteomics help in identifying antigen can trigger an immune response, which is vital for vaccine design. By studying this proteome of pathogens like previously described, analyze the Mycobacterium tuberculosis or Plasmodium falciparum, Now, for researcher can identify what potential vaccine candidates. These are all about the proteomics, genomics approaches. तो आज क्लास पूरा टोटल हम कोर्स जहाँ थिला हमें सब कवर कले जदि कोन अछि डाउट को
सर क्वेश्चन केमती आसीबो टिके क्वेश्चन किछटा करी करी पठेले हमें टिके प्रिपेयर करंतु
हम
किछ
पचारि
आईडिया नहीं क्वेश्चन केमती आसी
ठीक अछि कारण तमर तो एटा बदे फर्स्ट टाइम होछि कि कोन मु
सर नॉर्मली पीजी पिलांकर जेमते क्वेश्चन आस्तु हमर भी तो से ही आसीबो ना
हां
ना
सेमिति
डिफरेंट हाँ तो आपण टिके
मु
सेईटा टिके
पैटर्न
जदी हेल्प
जानिनी
करबो काहे बहुत
जदि
कष्ट टोटल ये एतिको पढ़िया एकना तो क्वेश्चन हेले प्रिपेयर कला बेलको 20 टा खंडे क्वेश्चन जदी हम प्रिपेयर कर दू मे बी हे जिवो सेमती भाबुछु से सटे हेल्प करंतु सेटा
ठीक अछि मु पचारि कि सर कहबी जदि कोन तांगर पैटर्न थिबो ताले मु जानिले मु इन्फॉर्म कर नले
ठीक
आपण
है
जे कोऑर्डिनेटर अछंती
आज्ञा
आपणकर जे अछंती कोऑर्डिनेटर अछंती कि डायरेक्टर अछंती आपणकर सर तांको पचारंतु जे पैटर्न कोन केन टाइप ऑफ क्वेश्चन पैटर्न तो सही टाइप रो आपण ये करिवे मु भी पचार देथिबी जदि किछि पैटर्न मु जानिली ताले आई विल इन्फॉर्म टू यू
आज्ञा आज्ञा
ओके
थैंक यू सर
आ जदि कार जदि डाउट रहिबो कि किछि रहिबो त मते पचारि परिबे किछि असुविधा
यस सर
जदि किछि न बुझि
सर
पारले ताले मते पचारि परिबे
ମୁଁ ଖାଲି
सर
ଡାଉଟ୍ ରେ କିନ୍ତୁ
एक्चुअली
ନୋଟ୍ ମାନେ ଏତେ କମ ସମୟ ଭିତରେ ନୋଟ୍ ବି ତ କରିହବନି ସାର୍ କେମିତି ସେଟାକୁ ଆମେ ମାନେ
ଆପଣ ଦେଖନ୍ତୁ ମୁଁ ବେସିକ ଯାହା ଥିଲା ମୁଁ ପଢେଇଲି ତ ଆପଣ ସେଇଟାକୁ ଯାଇକି ଆପଣଙ୍କର ଯୋଉ ଡିଫରେଣ୍ଟ ବ୍ଲକ ନାହିଁ କି ସେଇଟାରୁ ଥରେ
ଆଜ୍ଞା
ରିଡ୍ ମାରନ୍ତୁ ସେଇଟା ଆପଣଙ୍କୁ ସେଇଥିରୁ ନୋଟ୍ କରନ୍ତୁ ମୋ ପିପିଟି ଦେଖନ୍ତୁ ସେଇଥିରୁ ନୋଟ୍ କରନ୍ତୁ ଆଉ ଯଦି
ଆଜ୍ଞା
କିଛି ରିକ୍ୱାରମେଣ୍ଟ ରହିଲା ତ ମତେ ମେସେଜ କରନ୍ତୁ ତ ଆଇ ୱିଲ୍ ସେଣ୍ଡ ଦ ନୋଟ୍ସ
सर एक्चुअली ओवरऑल तो आम बुझीगलु माने आपण जहाँ आपनको क्लास गुड़ाक किसी प्रोब्लेम हबनी कि पढ़िला बेले सर गोटे गोटे वर्ड किंबा गोटे गोटे जोटा हम पई नुआ
ଠିକ
हबो
ଅଛି କିଛି
सेटा
ଅସୁବିଧା ନାହିଁ
टिके
ମତେ
डाउट
ପଚାରିବେ
रहइबो आ
ନା ନା ପଚାରି ପାରିବେ ଆପଣ କିଛି ଅସୁବିଧା ନାହିଁ।
यस सर
ଯୋଉଟା ବି ଡାଉଟ ରହିବ ମତେ ପଚାରି ପାରିବେ କିଛି ଅସୁବିଧା ନାହିଁ
ओके सर
ଯାହା ଡାଉଟ ରହିବ ମତେ ପଚାରିବେ ଆଉ
थैंक यू सर
सर सर जो हमर रेस्ट आउ जते क्लासेस गुडा थिला जो पारासाइटोलॉजी रो ताकू सर आउ केबे कोन होबो
ସେଇଟା
ता
ସିଏ
क्लासेस गुडा
ଇନଫର୍ମ କରିବେ ଆପଣ ମୁଁ ଇନଫର୍ମ କରିଦେବି ଆଉ ସାରଙ୍କୁ ପଚାରିକି ଗୋଟେ ସ୍ଲଟ ନେଇକି ଯଦି ହବ ମୁଁ ପଚାରିକି ଇନଫର୍ମ କରିଦେବି ଆଉ ଠିକ ଅଛି
आगे
ओके सर थैंक यू
ତମର ତ
सर
ଏବେ କଣ୍ଟିନ୍ୟୁଅस କ୍ଲାस ଚାଲିବ ନା
आगे
हाँ हाँ सर
ହୁଁ ଠିକ ଅଛି
सर
ମୁଁ
आउ किछि क्लासेस आपन को सर जदि हम रिक्वेस्ट करू सर आपन को ताले से कोन कोऑर्डिनेटर रिक्वेस्ट कले से कोन आपन को आउ किछि क्लासेस देबे जेमति कि नेक्स्ट किछि आउ किछि क्लासेस पे आपन को
ହଁ ସିଏ ଯଦି ଆମକୁ ଇନଫର୍ମ କରିବେ ଦେନ୍ ଆମେ ଦବୁ କିଛି ଅସୁବିଧା ନାହିଁ
सेटा रीजनल सेंटर रे कहले हबो से माने अलर्ट
ହୁଁ
करिवे
ସେଟା ରିଜନାଲ ସେଣ୍ଟରରେ ସେ ଯଦି କହିବେ ତାଙ୍କର କଣ ପ୍ରୋଭିଜନ ଥିବ ଟର୍ମସ ଆଣ୍ଡ କଣ୍ଡିସନ ଥିବ ମୁଁ ଜାଣିନି ସେଟା ସେମାନେ କହିବେ ଠିକ ଅଛି ଆପଣଙ୍କର ଯାହା ଡାଉଟ ହବ କିଛି ନାହିଁ ମତେ ମେସେଜ କରିପାରିବେ ମୁଁ କହିଦେବି ଆପଣଙ୍କୁ
सर
ଯାହା note
सर
यस
को
सर
टाइम रे मैसेज सर सर टाइमिंग टा गोटे जोनंती सर जो टाइम रे हमें
नहीं
आपन
नहीं मेसेज
रेगुलर
करे
हमें आपन
टाइमिंग
मैसेज
कोन
कर
सार जतले
ମେସେଜ
देखिबे कि स्लाइड
ମେସେଜରେ
दा
କିଛି ନାହିଁ ଆପଣ ମେସେଜ ଡ୍ରପ କରିଦେବେ ମୁଁ ପଢିକି ଯେତେବେଳେ ମୋ ଫ୍ରି ଟାଇମ ଥିବ ମୁଁ ଆପଣଙ୍କୁ ପଠେଇଦେବି
आगे
ଠିକ ଅଛି ହୁଁ। ଠିକ ଅଛି ଆଜିର ସେସନ କୁ ଆମେ ଏଇଠି କ୍ଲୋଜ କରିବା ଆଉ ସବୁ ସରିଲା ସେଥିପାଇଁ ମୋ ପାର୍ଟରେ ତମମାନଙ୍କୁ ଅଲ ଦ ବେଷ୍ଟ ତମମାନେ ପଢ ଯଦି ଡାଉଟ ହେଲା ମତେ ପଚାର ଠିକ ଅଛି
सर
यस
आपन को
सर
सर आपन को बहुत बहुत धन्यवाद सर बहुत भल्ले रे सर आपण टॉपिक सब गुडा कवर कले ଏଇଟା ବୋଧେ ଆମର ସବୁଠୁ ବେଷ୍ଟ ରହିଲା ଆପଣଙ୍କର ଯେତେଟା କ୍ଲାसेସ୍ ରହିଛି ସବୁ ଆମେ ଭଲସେ ବୁଝିପାରୁଛୁ। ମୁଁ ଭାବୁଛି ମୁଁ କଣ ସବୁ ପିଲା ବି ସମସ୍ତେ ଭଲସେ ବୁଝିପାରିଥିବେ। ଆପଣ ସାର୍ ବହୁତ ବହୁତ ଧନ୍ୟବାଦ ସାର୍।
ସାର୍ ବହୁତ
Thank
ବହୁତ
you,
ଧନ୍ୟବାଦ।
thank you.
Thank you, sir. Thank you.
Thank you very much, sir.
Thank you, sir.